Hello, dear students. I'm sorry due to interruption, we couldn't continue that one. Now it's continuation of the uh, enzymes, particularly the role of enzymes in the germination of the seeds. Uh, the soluble product of digestion are translocated to the plumule and radicle. Glucose is used for tissue respiration to provide energy for cell division and growth, to form new protoplasm and to make cellulose for new cell walls. The amino acids are used in the formation of new cell proteins in the plumule and radicle. Now, how enzymes work? For that is a special hypothesis is used, which is called as the lock and key hypothesis. According to this hypothesis, the job of an enzyme, for example, those used in digestion, is often to break down a large molecule, the substrate molecule, into smaller molecule, the product. The lock and key hypothesis suggests how this may be achieved. Each enzyme in a molecule with a specific shape on part of its surface is the active site, the lock, a section where its substrate molecules, the key fits. Exactly. So, my dear students, in this process, the enzyme will always act as lock and the substrate will act as key. And over the enzyme, there is a special site with which the substrate can be fixed. Uh, that is called as active site. Now, when the substrate molecule is in a position in the active site, the enzyme slightly stresses or bends the substrate, splitting it into two product molecules. The product molecules drift away from the enzyme molecule, leaving its active site free to operate again. So we have already told you the enzymes can be used again and again. In this way, when here in the diagram A and B are the substrates, they come close to the enzyme molecule, then they will make enzyme substrate complex, and then the enzyme will drift the substrates into the product. So a new product will be formed and then the enzyme will become free to be used again. So this was the synthesis enzyme. In the second diagram, we can see the enzyme molecule and a substrate molecule, which is yellow colored, blue colored enzyme, and both of these enzyme and substrate combine together to make a enzyme substrate complex. After that, the substrate is changed into the product and the enzyme becomes free. So this is the best example of lock and key hypothesis. Now, lock and key hypothesis explain enzyme action because only the correct enzyme substrate combination can work. Higher the temperature makes enzymes and substrate molecules move more quickly. This also means the substrate molecules enter the active site of the enzyme and the product molecules leave the active site of the enzyme more quickly. Number three, extreme heat causes the atoms of the enzyme molecule to move about so violently that they change position relative to one another. This changes the shape of the active site of the enzyme, stops doing its job. Now, the change in pH may alter the shape of the large molecule like proteins. If the protein is an enzyme, the shape of the active site may be changed and the enzyme will work less efficiently. By including a molecule of water in this process, the newly exposed ends of the product molecules are sealed, so they will not rejoin. This type of enzyme control reaction, common in digestion, is known as hydrolysis. So where the water is added is called as hydrolysis, and where the water comes out is called as condensation reaction. So condensation is a process in which smaller molecules join to form the larger molecules and water is released. Now the last topic of this main topic enzyme are the uses of enzymes. So enzymes are cheap to use in the industry. They do not need high temperature to work, so this reduces the cost of the fuel in the industry. Enzyme can be used again and again. This means they are only needed in small amounts. Proteases are used to tenderize the meat and to remove hair from the skin. Enzymes from microbes are used in fermentation to make beer, wine, and vinegar, etc. And carbohydrates are used in making chocolate, syrups, and other food products. Enzymes are used in digestion of food, enzymes are used in the germination of seeds, and enzymes are also used in clinical laboratories and extraction of juice from the fruits. So, my dear students, with this, the chapter is finished about the enzymes. So, here we quickly go through the chapter that is characteristics of the enzymes, about 8 to 10 characteristics like specific, catalyst, biocatalyst, made up of proteins, temperature sensitive, pH sensitive, water soluble, follow lock and key hypothesis, and have special site called active sites. Then their lock and key hypothesis should be explained more, and then uses of the enzyme. So the chapter can be summarized in this way. Okay, best of luck. Allah Hafiz.